Imagine if we could save all these critically endangered species, like the California condor or the northern white rhino. We don't know how to grow their cells in the lab and make them survive and thrive. Why is this so challenging? The truth is, each species plays by its own biological rules. Let's take birds, for example. The specific requirements for culturing cells can vary quite a bit, depending on which species we're talking about. The answer could be in this new study. They use primordial germ cells, or PGCs, which are the starter cells that eventually become eggs and sperm in birds. This is what makes PGCs a critical resource for conservation. To better understand the growth requirements for PGCs, the scientists collected samples from rock dove embryos. Sounds fancy, but really, pigeons. They processed them using a single-cell RNA sequencing method called PIP-seq. The team identified a distinct population of PGCs using their unique gene markers. They then looked at which growth factor receptors the cells expressed to figure out which signals they could respond to. So they found that if you add in growth factors IGF, FGF, and EGF, pigeon PGCs could be cultured for over 2 months and stayed healthy. This is a really cool way to use single-cell sequencing. This approach, using PIP-seq, can then be applied to things like conservation or biobanking. We can also figure out how to culture all kinds of different cells from different organisms, like the critically endangered axolotl. Because if there's one thing the world doesn't need more of, it's pigeons. But we thank you for your service.